Hi everyone, in this video, we will learn single-stranded RNA bacteriophages. So far, we have studied bacteriophages which have DNA as their genetic material such as T4, T7, lambda phage, then even single-stranded DNA phages such as 5X174, M13. But here, we'll be studying RNA bacteriophages. Yes, bacteriophages that have single-stranded RNA as their genetic material. Several single-stranded RNA phages have been isolated and studied. Example, E. coli phages F2, R17, MS2, Q beta. All these bacteriophages have single-stranded RNA as their genetic material. Each of these is a tailless icosahedron. That means these bacteriophages doesn't have a tail, they have an icosahedral capsid like 5X174. And these are the phages which infect bacterial cells having conjugative plasmids, and they infect bacteria by attaching to the pili encoded by that plasmid. The RNA is a single-stranded linear molecule. It's a single-stranded genetic material, linear. It has a great deal of intramolecular hydrogen bonding, which we'll see in the life cycle, and it consists of about 3,600 nucleotides. Now, for many years, we used to think that these single-stranded RNA bacteriophages have only 3 genes, but MS2 phage, it's very well known now, has 4 genes. One is the gene that encodes for the coat protein or the capsid, CP. Another is the attachment protein A, also called as maturation protein. Third gene is the RNA-dependent RNA polymerase or the replicase for replication. So the capsid gene for the coat protein or capsid, maturation or attachment protein, and RDRP. These 3 were known. But in MS2, there is also a gene that codes for the lysis protein, for the lysis of the host. And this is a very interesting feature because the gene for the lysis is an overlapping gene. It overlaps both the coat protein gene and the replicase gene, which you can clearly see in this case. The RNA molecule is both a replication template and mRNA because it's a plus strand RNA, which we'll see in detail in the life cycle. The burst size of these single-stranded RNA bacteriophages is very large, that is 5,000 to 10,000 compared to burst size of DNA bacteriophages. And the lysis of a bacterial population occurs gradually from 30 to 60 minutes after infection. Let's see the life cycle of a single-stranded RNA phage. The life cycles have been elucidated by comparing different features discovered from each of the single-stranded RNA phage, MS2, Q beta, and other phages. First step is entry to the cell and binding to ribosomes. So these phages attach to the host cell by binding to the pilus, and then they inject their genetic material, which is single-stranded RNA. And once the genetic material is injected inside the host cell, this genetic material is single-stranded RNA of positive polarity. What do you mean by a plus strand RNA? In a plus strand RNA, the base sequence of this genetic material or the plus strand RNA is similar to the base sequence of mRNA. In other words, the genetic material itself is the mRNA. So these viruses doesn't have to do the transcription. Their genetic material is already mRNA. They have ready-made mRNA, and after entry into the cell, this genetic material plus single-stranded RNA can be translated into protein. So the first step is entry to the cell and then binding to the ribosome. Immediately after entry, since this genetic material can directly be translated into protein, a ribosome attaches to the binding site at the beginning of the coat protein or the CP gene, which is the centermost gene in the RNA molecule, which you have seen just few minutes back in the genetic map. Next is translation and regulation of the amount of each of the 3 proteins or 4 proteins. Because for many years, people used to think it codes for only 3 proteins, but in MS2, as I said, there is a protein, lysis protein as well. So how these bacteriophages regulate the amount of each protein, because they don't need all the proteins at the same level. The ribosomal binding sites for the maturation protein, A protein, and the replicase are blocked in the R17 single-stranded RNA phage. This was known. So why the ribosomal binding sites for the A gene and the rep gene are blocked? By the presence of the secondary structures in the RNA stem and loop structures. So the A protein cannot be easily translated, nor can be the replicase. That's why when the ribosomes bind, they bind at the CP gene and they translate the coat protein. But translation of the CP gene or the formation of the coat protein opens up the binding site of the rep protein as well, and it also gets translated. So when the ribosomes bind, the A site or the A gene is blocked. The rep gene is also blocked because of secondary structures of stem and loop structures, but only the CP gene or the coat protein gene can be translated in the beginning. But translation of the CP gene opens up the binding site for rep so that the rep is also made, which is the replicase. But after some time, increasing amounts of the coat protein bind to the rep site and block the translation of the rep gene. So you would say that initially only the coat protein would be made. Yes. Rep protein and A proteins cannot be made because of stem loop structure. Yes. But the translation of the CP gene or formation of the coat protein opens up the site for the rep protein as well in the RNA so that it can also be translated and made. Yes. But later again, the CP protein only will go and bind to the rep site in the RNA to block the translation of the rep gene. Reason is that the replicase is no longer needed more, or I would say in very large amounts. This inhibition is efficient because the bacteriophage needs the coat protein in large amounts. About 2 into 10 raised to 6 copies of the CP protein are needed. When I told you the burst size few minutes back, I said it's very large as compared to DNA phages. The burst size is 5,000 to 10,000. So when a single RNA phage infects one E. coli cell, after the cycle is complete, the 10,000 phages would come out of a single E. coli cell. So the burst size is 10,000. One phage will infect one E. coli. After the cycle is complete, it will give rise to 10,000 phages. So for so many phages to be made, lot of coat protein is needed. 2 into 10 is to 6 as structural components, whereas the replicase is needed only in catalytic amounts. That's why the CP protein only stops the translation of replicase. It's needed only in catalytic amounts, but the coat protein synthesis continues because a lot of capsid is needed for 10,000 phages. So this is regulation of the 3 proteins or at translational level. Next is replication of the phage RNA. After the replicase enzyme is made, only the Q beta bacteriophage replicase has been studied in detail. I told you that this is a generalized life cycle of a single-stranded RNA phage, where they've combined the features of each of the single-stranded RNA phage, such as R17, MS2, and Q beta. So when it comes to replicase, only the Q beta replicase has been studied in detail. It's a tetramer consisting of one rep molecule, which is of course coded by the phage. You have seen the gene, and the other 3 units. When I say tetramer, that means it has 4 subunits. One is rep, which is coded by the phage. The other 3 are the host or the E. coli proteins, which is your subunits involved in the translation or elongation of translation of protein synthesis, EFTS, EFTU, and the ribosomal subunit S1. When the rep coded by the phage combines with this host proteins EFTS, EFTU, and S1, you get the complete replicase or the RDRP, and the replicase copies the plus strand, generate a minus strand. So you can see here the black strand is the plus strand of the virus, and from that the purple strand, what you can see is the minus strand. So black is plus, purple strand which you can see here is the minus strand. So minus would be made from plus. Now the minus strand is in contact with the plus strand only at the polymerization site. So yahan pe bhi you can see the minus strand is in contact with the plus strand only at the polymerization site. So the replicative form is single stranded. So the replicative form is single stranded, and also one more thing you can see here is that many minus strands are being synthesized before the completion of the first strand. So several minus strands synthesis occurs before the first minus strand is complete, which you can see here that this minus strand is being made, and before it's completed, the synthesis of other minus strand also begins. That's why the replicative form is a branch structure. Okay. After these minus strands are released, this minus strand is being denoted, or I would say shown in purple color here. After these minus strands are released, they will again be used as a template for the synthesis of the plus strand. The reason is the genetic material of these phages is single-stranded RNA. They cannot do replication in a single step. And when this plus strand RNA would be used as a template, the complementary strand obviously would be minus, because the replication cannot take place in one step. Now this minus strand cannot be packed into the capsid because that's not the genetic material of the virus. The genetic material is plus. So this minus is just a replicative intermediate or an antigenome which will be used as a template for the synthesis of more plus strands. So the minus strands are released and immediately used by the same replicase enzyme to form the plus strand, which you can see here. Purple is minus, and from that the plus strand would be made. So it's like that. This is the plus single-stranded RNA, the genetic material of these phages, MS2, Q beta, R17, and the replicase, which combines with the 3 subunits of host EFTS, EFTU, and S1, use this plus single-stranded RNA as a template to synthesize the replicative intermediate or the antigenome, which is minus single-stranded RNA. The replication is not complete here. This minus single-stranded RNA would be used as a template by the same enzyme to make the plus strand RNA. So this minus strand will be used as a template by the same enzyme to make the plus strand RNA, which is the genome. Now this plus strand RNA can be packaged into the progeny, and it can also go back for the translation to make more protein. So some of the plus strands return to the ribosomes for the synthesis of more coat proteins, and others are packaged. All progeny contain plus strands exclusively because that is the genetic material. The virus cannot pack minus strand by mistake. Before and once the plus strands are packaged into the icosahedral capsids, now they are ready to be released. But before we talk about assembly of viruses and lysis, we'll talk about synthesis of A protein. So I told you that in the beginning, when the ribosomes translate, coat protein is made, then replicase is also made, but the synthesis of A protein is not yet done. So synthesis of the A protein. Now the ribosomal binding site for the A protein is never available on a free plus strand because of the secondary structures. You would say that the genetic material of these phages is plus strand RNA. It can directly be translated. What's the big deal? But because of the secondary structures, the ribosomal binding site where the ribosomes can sit for the A protein or A gene is never available on a free plus strand. And this is the reason that the A protein is not synthesized very freely. However, replication of a minus strand begins at the 3' terminus adjacent to the A gene. Just after the plus strand synthesis begins, there is a brief period during which the ribosomal binding site for the A protein is free. During this time, the A gene is translated. The number of A proteins is maintained roughly equal to the number of plus strands. Jitna plus strands, utna A protein. This is economical. This arrangement is economical since each virus particle contains only one A protein molecule. Each A protein later becomes bound to the plus single-stranded RNA at an unidentified binding site. It's thought that the A protein facilitates the interaction of the RNA with the coat protein molecules and helps in the packaging. A protein remains bound to the RNA and enters the cell in the next cycle or subsequent infection. And after the A protein is also made and then the single-stranded RNA is packed, that's assembly. So molecules of the capsid or the coat protein spontaneously aggregate around the newly synthesized plus strand and form an icosahedral shell. Then lysis occurs after about 10,000 phage particles have formed because the burst size is large. Now this lysis gene, which I had shown earlier that it's a overlapping gene. Now this lysis gene, which is overlapping with some part of the CP gene and the RDRP or the rep gene, the start codon of the MS2 bacteriophage lysis gene is not easily accessible to ribosomes. The start codon of the lysis gene is not easily accessible because of the secondary structure of the RNA. When the ribosome terminates the synthesis of the coat protein gene, the secondary structure of the RNA is disrupted, and this disruption allows the ribosome to begin reading the lysis gene, and the lysis protein will be made. This low level of translation prevents premature lysis of the cell because the phage would not like to kill E. coli when only 100 phages or 200 phages are made. Then the phage would have been called as an unsuccessful phage. The phage would wait till 10,000 phages have been made. This is the reason that the lysis gene is not easily translated. The start codon is not accessible due to secondary structure. But when ribosome terminates the CP gene, the secondary structure is disrupted, lysis protein is made. But this low level of translation or regulation prevents the premature lysis only after sufficient copies of the coat protein are available for the assembly of mature virions. Thus, the cell lysis commences. So in this recording, we have studied single-stranded RNA bacteriophages, examples, genetic map, properties, and the life cycle and replication in detail.